Okay, well, I'd like to thank Shiro for the very kind invitation to come and present at this conference. I found it really fascinating. I can also tell the AV guys that I haven't got any audio running on my notebook, so you don't have to run around for 10 minutes trying to figure out what's going on. But it was lovely music, I'd have to say, David. So I hope that by the end of my talk I can convince you that there are still some things beyond what David's just described that can be done with imaging data. I was starting to wonder as I was sitting there watching it, but I think there are. And I guess my talk will be somewhat complementary in that at the end I'll really describe application of some of those connectivity approaches into a clinical neuroscience group, a Huntington's disease patient group, and using that as I guess a more neuroinformatics-based technique to be more specifically investigating parts of the striatum. But initially I'll start by showing you just really a simple view of our neuroinformatics or neuroimaging informatics system that we've built over the last 3 or 4 years. I'll then tell you how we use that in our basic neuroscience work, in fact with a motor task. That's already published, but I'll just briefly summarise that for you. And then how that really stimulated our interest to try and then investigate motor dysfunction and progressively more dysfunctional motor activity in Huntington's patients, as well as a number of other Mendelian disease patient groups. So our approach, which in a way was very simplistic, because at the time we did this about 4 years ago, we were trying to tap into some of the other efforts internationally with data models, and we found it challenging to pick up on another, if you like, standard of a data model. We weren't even thinking about ontologies at that stage, but we were conscious that we needed to have a way of describing our data, and I guess more particularly how we then were planning or in fact were processing our data at that time. So one of the things that was part of our agenda was to try and develop a way of incorporating the method into our data model. And so we then called it a data model with a method object, and so we ended up calling it a data object model, and ultimately called it a framework because it tried to incorporate the data, processing steps to the data, and storing obviously the metadata associated with the raw data, but then the metadata associated with the processing steps. And in particular, because we were in a sort of research environment which is based either on clinical neuroscience patient groups or animal models of diseases, we have I suppose a subject-oriented data model, and we aggregate subjects into projects and sometimes reuse subjects within different projects, and so we have a separate identity of the subject outside of our particular project. It was, I guess, sort of a step out into the dark for us, so we actually partnered with a small informatics company in Melbourne led by Jason Lowry, and it turned out that was strategically a really critical step. He's really a digital asset management guru and has just continually driven the engine underneath of what we were trying to construct on top for the purposes of neuroscience research. I've been sitting here listening to Marianne for the last 2 days, and I'm absolutely convinced we need to have an ontology in this system, this framework, and it's definitely coming. And hopefully next time I present I can show you the ontology, which is sitting up here somewhere above it. So that's obviously one of our short-term goals now. Here's just a couple of windows into how this looks for a user. There's obviously a whole file system that they can access. There's all sorts of authentication systems which we have to worry a lot about, because in our environment in the university we actually aggregate data, human imaging data from a number of hospitals, and of course that raises the issue of privacy of individuals' identities. So we've had to anonymise the data coming in from the hospital scanners, and we then have to of course have strong security layers for people to access this database. We've envisaged how we can share this data primarily with our collaborators who would have some sort of interoperable system, and one of the aspects we've worried a lot about is how we can provide secure access to parts of our database. So in fact each project has a separate security sort of wrapper, if you like, around that. We have obviously a range of different windows that show different experimental animal models. As you can see here, a couple of them are listed, different strains and other sort of obvious metadata that you'd like to have in there. We have some views then in terms of different studies, because there's a concept here of having longitudinal studies in these subjects, be they humans or animal models. So we have multiple series, and then of course other elements that relate to time-changing information related to different points where data is collected on the particular subject. As the data comes in, people can then check it out by visually inspecting it, or they can download it through the browser to their desktop, or they can have it transported to other sites around the university. And they can also just do some quick visual inspection, visualization of thumbnail images to know that they're looking at the right data, and to check through a lot of information from the header of the MR scans that are required. So this is a mouse model. It's an optic nerve study in an EAE model of multiple sclerosis. So that was just another example of how we're using this type of informatics system. Stepping up a level now, we were conscious that this sort of data, very well managed, sitting in a nice database, is of course critical, but the research advantage then becomes in how we can actually start to pipeline through analysis procedures on this data set. And in that context, we took a slightly different strategy based primarily on the PhD work of Eugene Duff, who was in my lab at that time and now is at Oxford. And he developed a tool called REX, or a regional exploration tool, which is really designed for functional MRI data sets. And so he then took on board through a MATLAB interface an ability to index all of the different types of MR scans for different subjects, different sessions, different conditions, and then actually data time points, so that one could then, through an indexed set of access to these types of data elements, be able to construct subsets of time series data for example, different sessions for a subset of subjects, or between patients and controls, or whatever. And so this enabled us then to have a visualization tool beyond what we'd previously, I guess, had the ability to look at, and that's really highlighted here, where you can see a whole series of time courses taken out of a particular session and then across a number of different sessions, and one can visualize very quickly for a region, which is a cluster of voxels within this 4D data set, the whole time series or subsets of it which relate to different tasks. And then one can ultimately build up, if you like, summation profiles of all of the different particular responses to repeated presentations of a stimuli or a motor task output, as was the case here. And then obviously ultimately generate some mean time course profile and a variance estimate around that based on this sort of high-level view of functional imaging data. So that, I guess, was our entree into trying to investigate the complexities of dynamic motor function within human subjects. And once we were doing this, it became apparent to us that it's actually a very rich data set. And in fact, that was already well known by others who had looked at, for example, here, a study by Harms 5 years ago, which showed the complexity of different types of primary and secondary auditory cortex responses to different auditory stimulation, and showing how it's clearly not just a single type of bold signal response within primary or secondary auditory cortex, but depending on the type of stimulus, there's I think a really terrific diversity of the bold signals. And so that was something that piqued our interest to look and see what we could look at with this type of informatics system in terms of motor function. This study was done in collaboration with Peter Fox and colleagues in Texas, and it was an experiment looking at motor learning, in fact, where people had to use their non-dominant hand, and they trained on a task which was a thumb and finger opposition task, touching on the first, third, second and fourth finger, and training in this task over a period of weeks and comparing that to an untrained controlled task. And in fact, what I'll show you here is just the baseline scans and results, where people were essentially, we were looking at the complexity of their motor function in the initial time point before the training effects are really evident. But the point of this actually is to be able to show you that over a continuous task period of 2 minutes, which was one of the rather novel aspects of this experimental design, we could see this full glory of the bold signal dynamic complex signal, and then had some insights into how we might be able to use this in some of our clinical neuroscience studies. And so this just shows you an output of the motor cortex here. This is shown in radiological format, so that's the right hemisphere on the left side of the image, and this is the classic M1 output that one would expect, with a little bit of extra signal, a small increased amplitude right at the stimulus onset, and when they've stopped the motor task, this small undershoot, which is a well-known phenomena in terms of the bold signal. But this here shows you other brain areas, so the region ipsilateral to the hand with their initial output and then some very low level of signal across this 120-second period. Other brain areas of interest such as mid-cingulate, obviously supplementary motor area here shown with essentially ramping increasing signal, and then other brain areas such as the dentate, the right dentate. So again, contralateral to the hand, and you can see here a signal that's nicely elevated but typically slightly decreasing throughout that 2-minute period. And what we could see is, in fact, there's a whole range of different types of bold signals which are evident in different brain areas, and that's what this deconstruction, or if you like, this basis set, it's a physiological inspired basis set to look at types of standard fixed boxcar responses in the brain, and that's the obvious areas we see here in the right motor cortex and SMA, for example, down here in the dentate. Here's the areas which have a constantly increasing signal, and that's mostly evident here in the SMA, and then other types of spiking onset, spiking offset, and then negative signals, in fact, as we saw in one of those earlier courses. So it really revealed to us that there's an enormous complexity in the bold task, and this was accessible to us by virtue of this informatics platform that particularly Eugene had built. So to try and get our heads around this, we went into another level of visualization, and with another colleague looked at how we could try and encapsulate the four-dimensionality of this bold signal data. So here's a mock-up of the actual real experimental data. You can see here that this schematic figure starts a left-hand activation. You can see this full expression of signal here in what is the right motor cortex, down here deep in the thalamus and in the left dentate. And as the task continues, you see that's a sustained level of activity, and then towards the end of the 120-second period, you see another flush of activity through the brain as you see various spiking phenomena that we could see in single time courses for single regions. So that gave us a window, if you like, into the complexity of the bold signal, and really piqued our interest to see how we could use this in some of the clinical neuroscience studies. And that became obviously very relevant to looking at motor dysfunction in Huntington's disease. And whilst we had the ability to do functional imaging, at this stage we were building on an initial structural imaging study, and we had added to that diffusion MRI, which David has just described to us, as well as tractography, to be able to in fact look at some of the paths and nodes within the motor network, and in particular the striatal areas involved in the motor feedback. So this is just a little bit of information on Huntington's disease. As we know, it's an autosomal dominant disease and has I guess a diagnosis which is really, whilst people are gene positive, individuals are not actually diagnosed until they have a sufficient severity of motor symptoms or psychiatric symptoms or affective symptoms. And this is something which is of particular importance because in terms of developing treatments for Huntington's disease, one has to think about how you would start a pharmaceutical therapy. At which time point would you start to administer drugs to individuals who were pre-symptomatic? So a driver in this research now has become to look at predictors or biomarkers of degeneration within the Huntington brain, and can you find the area and the measurement technique which is maximally sensitive to the earliest onset of neurodegeneration. And this is an example from a paper by Paulson and colleagues, which just highlights that whilst at some typical age of around 40 in Huntington's patients, or at least with groups having a CAG repeat length longer than 39, you can typically see a significant reduction into this clinical diagnostic motor threshold range and differentiate that from those with shorter CAG repeat lengths and clearly differentiated from control. And whilst that's very close to, in fact, the clinical onset or clinical diagnosis, we're interested to look back here as early as possible with brain imaging techniques to see, can we discern that there are degenerative processes occurring subclinically. And here's just a series of the circuits that we're interested in, most particularly here the motor and the spatial areas, particularly going through the putamen and the head of the caudate. So these are well-known areas of pathology in advanced Huntington's, and this study, which was conducted by a PhD student in my lab, India Bohanna, really particularly looked to see if we could identify these circuits with DTI to look at mapping the different parts of the striatum, the putamen and head of caudate, and associating those with potentially neurodegenerative measures with DTI, such as fractional anisotropy and mean diffusivity changes. And we have a group of controls and a fairly large group of pre-diagnostic Huntington's disease patients who are age matched with the controls, and typically in a range of about, I guess it's somewhere between 5 to 15 years pre-symptomatic or estimated years to onset. So this is the method we followed, which was first published by Tim Behrens and colleagues from Oxford. It's a beautiful technique. David mentioned it briefly, and this is just another view of how it works, where you're looking at the anisotropy of water diffusion in white matter tracts. You can characterise this with a single tensor model, which can then enable you to determine principal eigenvalues and principal eigenvector directions, for example, and then to try and use that in a colour-coded fashion, as David described, to look at different directionality of the preferred water diffusion in each voxel, and then in particular build up a model through the white matter of the brain where voxels are essentially connected with each other in terms of this preferred eigenvector direction or principal eigenvector direction. And Tim and colleagues showed beautiful parcellation of the thalamus using this technique where they essentially did thalamocortical tractography and used regions within the cortex to determine which were the nuclei in the thalamus that were tracking through to those areas of the cortex. So we followed a similar technique, and this is now in the case of looking at the caudate and the putamen, similarly mapping out the cortical areas, generating tracts. This is an example of a tract actually flowing from the thalamus up through the somatosensory cortex, but the same principle applies. And then using relatively sort of standard diffusion data now, which is 60 diffusion directions of a reasonable B-value, one can actually generate very nice maps of the connectivity from the cortex to the striatum in terms of brain areas and the head or the rostral part of the caudate, progressively through to premotor, more in the mid-section of the caudate, and down through into other regions such as posterior parietal or the temporal cortical connections through to this more lateral aspect of the head of the caudate. And here's the views of the putamen with similar mapping from cortical regions through to the head of the putamen or the rostral end of the putamen. And here's the sagittal view of how these different tracts were, parcellated out through this approach. And this technique, as I mentioned, was done in 14 subjects. These are controls, and we thresholded where there were at least 5 subjects that showed connectivity from those cortical areas through to the striatum. So this then is a summary of the findings, which shows the different cortical areas colour-coded to parts of the caudate here and the putamen. And of course the whole point of this then is to look in those areas of the putamen and caudate which have got specific tract or connectivity through to the motor cortex, because that's one of those key early symptoms in Huntington's disease. And you can see here that that's particularly this region here in the putamen, and there's a more medial view here, and up here in the caudate there's quite a reasonable representation. One particular surprising aspect of this finding was that, in fact, the head of the putamen is actually very strongly connected to the prefrontal areas, and that was somewhat surprising because, at least from the literature, our understanding was that it was primarily connected through to the head of the caudate from the prefrontal cortical areas. Anyway, the summary of this work then is to use this approach. So neuroinformatics approach is to parcellate out regions within the striatum. They're then plotted here across on the x-axis, and you can see in this pre-symptomatic group what we could see immediately in the caudate, in fact surprisingly to us, was that the posterior parietal area was the area that had the biggest initial volume loss. But when we then looked in our symptomatic Huntington's group, we could see that most of the regions through the caudate and quite a lot of the regions through the putamen had significant volume loss compared to controls. And then just two more slides which will highlight to you the ability to then look at more sensitive markers, and here I'm showing you the actual fractional anisotropy increase which occurs in the putamen in pre-symptomatic patients, and this is the mean diffusivity, which is also slightly increased, and in comparison to the symptomatic, where these effects become very pronounced through most of the regions of the striatum. So this is now the tool that we're using to explore in particular the premotor and sensory motor regions, M1 and S1, as well as the premotor and supplementary motor area. So here are the conclusions from this study. Firstly, you can do parcellation of brain areas with these very elegant tractography techniques. We've been able to investigate the connectivity and the changes of that, and in particular use that then to characterise diffusivity-based changes within this region of the Huntington's brain. Now, I'll skip over this slide, which is unfortunate because it's very visually appealing, but here's a little thought I would like to leave us with, Shiro, because sitting listening to the beautiful techniques which people have been presenting over the last couple of days, it reminded me of a quote that I once heard from T.D. Lee, the famous Nobel laureate in physics in '57, and I was fortunate as a graduate student to hear him give a lecture. Of course, he was a theorist, but he said that without experimentalists, theorists can tend to drift. And without theorists, experimental physicists can tend to falter. And that really stuck in my mind. I thought that's a really profound thought, and it's a really great stimulus for the interplay between experimental and theoretical work. But in my research, what I've found, I think in a similar analogous way, is that without brain and mind diseases, neuroscientists may tend to drift somewhat, whereas without neuroscience discoveries from basic scientists, I think brain disease cures may tend to falter. So I'd like to thank you again for the invitation, Shiro, and for your attention.
Thank you very much. Okay. Any question or comment? Yes.
So your DTI connection diagrams between cortex and basal ganglia are amazing. What do we do for verification?
That's a critical question for the community at the moment. I guess my personal view is that there needs to be a number of primate studies done, higher primate studies, and that would give us, I think, a significant degree of confidence and hopefully enough confidence. I mean, the other factor is empirically that when you use these techniques, and hopefully increasingly with individual subjects showing differences, more pronounced differences, that we'll see increasing correlation between these biomarker measures and the behaviour, if you like, of those individuals. Through a group of normal controls, I think it's much more difficult because the variability is so much less, but once we get into these types of diseases, it gives us that opportunity to get a stronger, if you like, confidence through better correlations.
Any other? Okay, thank you very much again.